Imagine you're trying to bake a cake, a really specific high-stakes wedding cake. You've got all the ingredients right there: flour, sugar, eggs. You have an oven. But the recipe you're using wasn't actually written for a cake. It was written for a pie. And it's in a language you only sort of understand. And to top it all off, half the instructions are just smudged out.
That sounds like a guaranteed way to ruin a wedding.
Exactly. And yet, for the last 20 years, that is essentially what some of the smartest scientists in the world have been doing when trying to cure diabetes.
Right. They've been trying to cook up human insulin-producing cells, beta cells, using a recipe that's, well, it's derived almost entirely from mice.
And while they've made some edible pies, I guess, they haven't quite nailed that wedding cake.
It's a classic case of close but no cigar. And in biology, close doesn't really cure diseases.
But today, we are diving into a paper that claims to have finally found the right recipe. We're looking at a study by Yuhad et al., published in 2026, titled Reconstructing Human Pancreatic Gene Networks Enhances Stem Cell-Derived Beta Cell Induction.
This is a huge paper. I mean, we aren't just talking about a small tweak in efficiency. We were talking about moving from what was basically a guessing game where we got, maybe 18% usable cells to a precision map that yields 70% pure functional beta cells.
So here is our mission for this deep dive. We're going to deconstruct this paper layer by layer. We need to understand the map, how human embryos actually build a pancreas versus how mice do it.
Then the error, why our current billion-dollar protocols are, kind of hilariously, making stomach cells by accident.
And finally, the fix. Protocol D and the stunning and a little bit counterintuitive results they saw when they actually put these cells into diabetic mice.
The core conflict here is really simple. It's the difference between making a cell that looks like a beta cell under a microscope and making a cell that actually functions and develops like a human beta cell.
Okay, so let's start at the beginning, the blueprint. Why have we been using the wrong map for 2 whole decades?
Well, it really just comes down to access. I mean, you can't exactly study human embryos in real time in a lab.
Of course not.
It's ethically and logistically, impossible. So for a long time, we relied on the mouse model. Mice are mammals. They have pancreases. We have pancreases.
The assumption was the developmental pathway was the same. A pretty safe assumption, usually.
Usually. But this paper just blows a hole in that. It exposes a critical discrepancy. In a mouse, the pancreas develops largely from a region called the midgut, or the dorsal pancreatic endoderm.
Okay, so if you're visualizing the developing gut as this long tube, the mouse pancreas mostly just pops out of the back of that tube.
That's it. So the mouse blueprint says, go to the back of the gut, start building the backdoor method, if you will.
Okay, the back door. But what did you and his team find in humans?
Well, they didn't just assume. They performed single-cell RNA sequencing, cRNA-seq on actual human embryos. We're talking Carnegie stages 10 through 15. This is incredibly early, like weeks 3 to 5.
And the human map, did it match the mouse?
Not at all. It was a completely different entry point. They found that humans don't really rely on that midgut pathway much at all. Instead, human pancreatic development is heavily driven by the anterior lip and the ventral pancreatic endoderm.
The anterior lip. So if the mouse is using the back door,
humans are using the front door.
And that's not just a small geographical difference, is it?
Oh, it changes everything because the chemical signals, the little traffic lights that tell cells what to become, are completely different at the front of the house than they are at the back.
So by using the mouse recipe, we were shouting instructions at the wrong door this whole time. But just knowing the location wasn't enough, was it? They had to understand the wiring.
Yeah. Which brings us to GCNs.
Right. Gene co-expression networks. And this is a really crucial shift in thinking.
And break this down for us. Why not just look for, individual genes?
Traditionally, that's what scientists did. Look for marker genes. It's like a roll call. They'd ask, is gene X present? If the answer was yes, they'd assume, okay, it's a pancreas cell. But just because I'm holding a spatula doesn't mean I'm a chef. I could just be some guy holding a spatula.
That is a perfect analogy. GCNs don't look at solo artists. They look at the whole orchestra. They look for modules, these groups of genes that turn on and off together.
I see.
If the violin section, the percussion, and the brass are all playing the same symphony at the same time, then you know you've got music.
So instead of just checking if a light is on in the building, we're checking if the finance department HR, and engineering are all in the conference room having a meeting.
Exactly. And when they mapped the natural human embryo using these GCNs, they found this incredibly clean, rigid structure. It wasn't a mess. It was organized into exactly 3 distinct modules.
The 3 modules. This is the gold standard they were aiming for. We have to visualize this.
Think of it as a 3-step ladder. Module US is the foundation. This is the early stage where the cell stops being a generic gut cell, the definitive endoderm, and commits to being a pancreatic progenitor.
Okay, and who's in charge there?
That team is controlled by transcription factors like FOXA1 and SOX17. They're the foremen on the job site.
Module 1, the foundation. Got it. What's next?
Then we have Module 2, the transition. This is where the cells differentiate into tip or trunk cells. The absolute boss of this module is a gene called PDX1.
PDX1. I feel like that's the one gene everyone in this field knows.
It's the celebrity. It's the master regulator. If PDX1 isn't working, you simply do not get a pancreas, period.
So Module 2 locks in that identity? And Module 3?
Module 3 is the destination. These are the endocrine lineages, the cells that will actually make the hormones like insulin. This network is controlled by NGN3 and Neurod1.
So in a human embryo, it's a simple 1, 2, 3-step process: foundation, transition, destination.
Correct. Clean as a whistle. And that simplicity is key. If you see anything else, something has gone very wrong.
Okay, let's do a quick recap here. We're about 5 minutes in, and we've established the gold standard, the target.
Right. So point 1, humans use the anterior lip, the front door pathway, not the mouse midgut back door.
Point 2, we have to look at gene co-expression networks, the whole orchestra, not just single genes.
And point 3, the most important one. The natural human pancreas is built on a strict, clean, 3-module architecture. To cure diabetes, we have to replicate that 1, 2, 3 ladder.
But, and this is the big twist, we haven't been. The paper then shifts to the diagnosis. They looked at the best protocols we currently have for making these cells in the lab.
Yep, they analyzed the 3 heavy hitters. They called them Protocol A, B, and C, but they correspond to the famous methods from Rosania, Hogrib, and Balboa. These are the gold standards everyone uses.
And when they ran these lab-grown cells through that GCN analysis, did they see the 1, 2, 3 ladder?
No, they saw a 4-module structure.
4 modules, so they, built an extra floor on the house?
It's more like a glitch, a structural defect. When they looked closely at this weird 4th module, they found it was packed with genes that had no business being in the pancreas.
What kind of genes?
Gut genes, gastrointestinal markers. We're talking about CDX2, PITX2, HOXA1. These are literally the instructions for making stomach lining and intestines.
Wait, hold on. So we're trying to make insulin cells, but we're accidentally making stomach cells.
Exactly. The final product was contaminated. Protocol A, for instance, only produced about 18% true beta cells. The rest was a mix of confused cells and these off-target gut cells.
It's like trying to drive to the pancreas, but you take a shortcut through the stomach and get stuck in traffic.
That is a perfect description of what was happening. And they pinpointed the exact cause. It wasn't just bad luck. It was the fuel we were using.
The fuel.
Growth factors, specifically a molecule called FGF10. In mouse development, you need FGF10 to push the cells along. So the old protocols were just dumping massive amounts of it, about 50 nanograms per milliliter, onto the cells.
15 nanograms. For context, is that a lot?
In this world, it's a fire hose. And combined with that, the protocols were rushing the timing. They were forcing the cells to sprint from stage 2 to stage 3 in just 2 days.
So we're shouting, go, 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 and just blasting them with this growth fuel.
And the cells got confused because they were rushed. And because of that high FGF10, they started having an identity crisis. They turned on the pancreas genes, sure, but they also turned on the gut genes.
They became these weird hybrids.
Polyhormonal, confused hybrids. They didn't know if they were supposed to digest food or regulate sugar. And a confused cell is a useless cell for therapy.
This explains so much about why stem cell therapy has been so hit or miss. We thought we had the right cells because they had some of the pancreas markers.
But deep down, their entire network was broken. The topology was just wrong. A 4-module network cannot function like a 3-module organ.
So we have the diagnosis, the gut problem. The cells are rushing and getting contaminated. Let's just pause and make sure we all see the issue.
Okay, a visualization check. Imagine a clean, pure stream of water. That's the 3-module pancreas. Now imagine a muddy tributary, which is the gut, pouring into it because we broke the dam with high FGF10. That's the old state of the art.
So how do we fix the dam? This is where the paper introduces Protocol D, the solution.
Protocol D is just beautiful because it's based directly on that first discovery we talked about, the anterior lip.
Right.
The researchers just asked, what is the chemical environment of the anterior lip? What does the front door smell like?
And what's the answer?
It smells like two things, Wnt and activin. These are signaling pathways. So step 1 of Protocol D, they changed the chemical bath. They added activin A and a small molecule called CHIR 99021.
CHIR. That sounds like a robot's name.
It's a chemical tool that powerfully activates the Wnt pathway. By adding these two, they're chemically telling the cells, you are in the anterior lip, you are at the front door.
Okay, so they corrected the GPS signal. But what about the rushing, the timing problem?
This is the most counterintuitive part. To get a better, more mature cell at the end, they had to slow down the process. They extended stage 2 from just 2 days to 4 days.
They doubled the time.
It's all about something called chromatin remodeling. To get a little technical, but it's fascinating. Think of the cell's DNA as a giant library where most of the books are glued shut.
Okay, I'm with you.
To become a pancreas cell, you need to unglue the pancreas books and, very importantly, keep the stomach books glued shut. This process, the physical opening and closing of chromatin, it takes time.
And if you rush it, you end up with a bunch of half-opened, messy books.
Exactly. You get a messy library. By extending stage 2 to 4 days, they gave the cells enough time to properly open the pancreatic genes like PDX1 and, crucially, to close the gut genes.
Let the cement dry before building the next floor.
And then the final stroke of genius. When they moved to stage 3, they cut the FGF10 levels.
They turned off the fire hose.
Drastically. They went from 50 nanograms down to just 2 nanograms.
From 50 to 2. That's a massive reduction.
They basically starved the gut pathway. Without that high FGF10 signal, the cells physically cannot turn on the gut genes. They are forced to stay on the pancreas track.
That's amazing.
Yeah.
It's not just encouraging the right behavior, it's making the wrong behavior impossible.
And they proved it using Gesundheit.
Gesundheit.
Yeah. Single nucleus ATAC sequencing. It lets you see which parts of the genome are open or closed. In Protocol D cells, the gut chapter of the DNA was sealed shut. The pancreas chapter was wide open for business.
And the yield. We were stuck at 18% before.
Protocol D achieves approximately 70% pure beta cells. And they're NKX61 positive and C-peptide positive, which means they are the real deal. High-fidelity, functional cells. The 3-module architecture was restored.
Okay, let's recap the fix before we get to the final proof in mice. This is the recipe you want to write down.
Right. Step 1, mimic the anterior lip with Wnt and activin agonists.
Step 2, slow down. Extend stage 2 to 4 days to let the library books get sorted out properly.
Step 3, starve the error. Cut FGF10 way down to 2 nanograms to stop that gut contamination.
And the result? A pure 3-module pancreas.
But, as we always say on this show, a cell in a dish is just a science experiment. A cure has to work in a living body.
Which brings us to section 4, the proof. They took these Protocol D cells and put them into mice.
Specifically, STZ-induced diabetic NOD SCID mice. These are mice with no functioning beta cells of their own. They are severely diabetic.
And I'm going to assume the cells worked.
They worked brilliantly. They normalized fasting blood glucose levels significantly better and faster than the old Protocol A cells. But there was a surprise in the data, a vascular surprise.
Ooh, I love a good surprise. What happened?
Well, usually in stem cell transplants, the logic is older is better. You want to transplant fully mature adult-like cells because you want them to start working right away.
Right. You don't send a trainee to do the job. You send the expert.
But the researchers found the exact opposite. They transplanted cells from different stages of Protocol D, and they found that the younger cells, specifically stage 5 cells and early stage 6 cells, performed better than the fully mature ones.
Wait, what? The trainees did a better job than the experts. Why?
Because the experts are rigid. Fully mature cells are kind of fragile. When you transplant them, the trauma of the surgery can kill them, or they just fail to engraft. They don't connect to the host tissue.
I don't like moving house.
Exactly. But the younger cells, they're plastic. They're adaptable. The researchers looked at vascularization, how blood vessels grow into the graft.
Using CD31 staining, I see.
Yes.
Yeah.
And they saw that the mouse's blood vessels connected to the younger grafts much, much faster. The cells were still developing, so they integrated into the mouse's body and then finished maturing inside the host.
That makes so much sense. It's like planting a tiny sapling versus trying to transplant a giant 100-year-old oak tree. The sapling root's better.
Precisely. Within 30 days inside the mouse, those young cells had finished their training and were regulating blood sugar perfectly.
That is a huge finding for actually getting this to clinical trials. It means we don't have to spend months growing these cells to perfection in the lab.
Right. We can get them started, put them in, and let the body finish the job. It saves time, it saves money, and it actually produces a better outcome.
And they showed it wasn't a fluke.
They tested Protocol D on different cell lines. They used the H9 line, which is notoriously difficult to work with, and also iPSCs.
Induced pluripotent stem cells.
And it worked across the board. The 3-module logic held up every time.
This is, honestly, it feels like a real turning point. We've gone from cooking by smell to cooking with chemistry.
It really is. It's a triumph of network analysis over just blind trial and error.
So let's bring this all home. We started with a broken recipe, translated from mouse to human, that was creating this confused slurry of pancreas and stomach cells.
By listening to the human embryo, specifically that anterior lip region, we learned we needed Wnt and activin.
We learned that rushing the process breaks the network. We have to slow down to let the chromatin remodel itself.
And we learned that by slashing FGF10, we can starve the bad outcomes and achieve 70% purity.
And finally, we learned that younger, more adaptable cells are actually better at curing diabetes in a living animal because they integrate better with the body's plumbing.
It's a complete overhaul of how we should be thinking about tissue engineering.
Which leads me to the big question I want to leave everyone mulling over.
I'm ready.
We fixed the pancreas by realizing we were using a 4-module map for a 3-module organ. We were fixing a network topology error.
Right.
So, what about the heart? What about the liver? What about the kidney?
That is the trillion-dollar question, isn't it?
How many other failed tissue engineering projects are sitting out there, just 1-module correction away from working?
It forces every biologist to go back and check their gene co-expression networks. Are your modules clean? Or are you accidentally making, I don't know, stomach cells in your brain dish?
It's time to double-check the blueprints.
And maybe stop relying so much on the mice.
Thank you for diving deep with us today. This is the kind of story that reminds you that sometimes, to go fast, you really do have to slow down.
Check your map.
See you on the next deep dive.